Welcome back. I hope you had some fun exploring the pre-processing code. And for this next lesson, we'll look into ERP experiments. So here's what we should already know. EEG datasets, including ERPs, can be accessible online from a variety of sources. And as a reminder, ERP is a technique in which neural responses to specific events are extracted from the EEG. Here's some key questions for this video. What are examples of ERP components? What are some famous ERP experiments? And why are ERPs useful? So first of all, I would like to cite and recommend this wonderful book about ERPs written by Stephen Locke. This is a great book if you want to learn more about ERP experiments. So before we take a look at some ERP experiments, let's first establish the naming conventions used for ERPs. So what we have here is a graph in the time domain with voltage values on the y-axis, and 0 milliseconds is the time of the stimulus onset. So this graph is the resulting averaged ERP waveforms that consist of a sequence of positive and negative voltage deflections, which are called peaks, waves, or components. So in this figure, the peaks are labeled N1, P1, N2, P2, and N3. So P and N are traditionally used to indicate positive going and negative going peaks, respectively, and the number indicates a peak's position within the waveform. So for example, P2 is the second major positive peak. Alternatively, the number may indicate that the latency of the peak in milliseconds after the stimulus. For example, the N170, which is a famous ERP component related to seeing faces, is simply here at around 170 milliseconds. And I'll show you the N170 peak later when I talk about the N170. If the component number is high or perhaps greater than 10, you should assume that it is referring to the peak's latency. So these peaks, P1, N1, P2, N2, and so forth, these components apply when the starting time is the stimulus onset. So this is why these naming conventions are specific to ERPs. So if you're watching this video in Google Colab, which I think doesn't allow full screen for YouTube videos, you can go ahead and watch this video briefly on YouTube so you can see this large image more closely, and then come back to the Colab whenever you're done watching this. So here's an example of a famous ERP experiment called the classic oddball paradigm, where subjects viewed sequences consisting of 80% X's and 20% O's. And they pressed one button for the X's and another button for the O's. So each letter was presented on a computer monitor for 100 milliseconds, followed by a 1400 millisecond blank interstimulus interval. So during each recording session, we viewed the EEG on the computer, but the stimulus elicited ERP responses were too small to notice within the much larger EEG. So figure C shows the EEG that was recorded at one electrode site from one of the subjects over a period of 9 seconds or 9,000 milliseconds. The EEG waveform shown in the figure was recorded in the PC electrode site, where the P3 wave is largest. So figure D is a blow-up of one small period of time, showing that the continuous voltage was converted into a discrete set of samples for storage on a computer. At the end of the recording session, we perform a simple signal averaging procedure to extract the ERP waveform elicited by the X's and the ERP waveform elicited by the O's. So the basic idea is that the recorded EEG contains the brain's response to the stimulus plus other activity that is unrelated to the stimulus. And we can extract this consistent response by averaging across many trials. So to accomplish this, we extracted a segment of EEG surrounding each X and each O, which is indicated by the rectangles in figure C. So these are the X, and this is the X segments, and this is like O segments, and this is these are another X segments. So, and we line up these EEG segments with respect to the event codes that mark the onset of each stimulus as shown here in figure E. We then simply average together the single trial EEG waveforms, creating one averaged ERP waveform for the X's and another for the O's at each electrode site, shown here in figure F. So for example, the voltage at 24 milliseconds in the average X waveform was computed by taking the voltage that was measured 24 milliseconds after each X stimulus and averaging all of these voltages together. Any brain activity that was consistently elicited by the stimulus at that time will remain in the average. However, any voltages that were unrelated to the stimulus will be negative on some trials and positive on other trials and will therefore cancel each other when averaged across many trials. Overall, the sequence of ERP peaks reflects the flow of information through the brain, and the voltage at each time point in the ERP waveform reflects brain activity at that precise moment in time. So that was the classic oddball paradigm. And this next example asks why behavioral reaction times are typically slowed in schizophrenia patients when they perform simple sensory motor tasks. That is, are reactions times slowed in patients because of an impairment in perceptual processes, or an impairment in decision processes, or an impairment in response processes? So ERPs are ideally suited for answering this question because they provide a direct means of measuring the timings of the processes that occur between a stimulus and a response. The researchers hypothesized that the slowing of reaction times in schizophrenic patients for simple tasks does not result from slowed perception or decision, but instead results from an impairment in the process of determining which response is appropriate once the stimulus has been perceived and categorized. And this is called the response selection process. As in many previous ERP studies, the voltage during the period of the P3 wave, which is approximately 300 to 800 milliseconds after stimulus onset, was reduced in the schizophrenia group relative to the control group. Researchers use certain techniques such as isolating specific ERP components by means of difference waves, in which the ERP waveform elicited by one trial type was subtracted from the ERP waveform elicited by another trial type. And this is similar to the difference images in fMRI studies. Difference waves are extremely useful in ERP research because they isolate neural processes that are differentially active for two trial types, eliminating the many concurrently active brain processes that do not differ from these trial types. So this is important because the different ERP components are ordinarily mixed together, making it difficult to determine exactly which component and which psychological or neural process differs across conditions or groups. So difference waves can pull out a subset of the components, making it possible to draw more specific conclusions. So in this study, the difference waves allows the researchers to better isolate the P3 wave and to focus on brain activity that reflects the classification of the stimulus. And from this, the researchers found that the slowing of reaction time reflects an impairment in processes that follow after stimulus categorization. Thus, for a relatively simple perceptual task, the slowed reaction times exhibited by the schizophrenic patients appear to result primarily from a slowing of response selection rather than a slowing of perception or categorization. So the next famous ERP study that I'm going to discuss is the N170 experiment, which is about face processing. So in your Python notebook, you will be looking at some basic data analysis of the N170 after watching this video. This experiment focuses on the N170 component, which is a face-related component that typically peaks around 170 milliseconds after stimulus onset and is largest over ventral areas of the visual cortex. In a typical N170 paradigm, photographs of faces and various types of non-face objects are briefly flashed on the computer monitor, and subjects passively view the stimuli. The N170 component here is significant because it is larger when the stimulus is a face compared to when the stimulus is a non-face object, such as an automobile. So the difference between faces and non-face objects begins approximately 170 milliseconds after the onset of the stimulus. This simple fact allows us to conclude that the human brain is able to distinguish between faces and other objects within 170 milliseconds. Many researchers have used the N170 component to address interesting questions about how faces are processed in the brain. So for example, some studies have asked whether face processing is automatic by testing whether the face-elicited N170 is smaller when the faces are ignored. The results of these experiments indicate that face processing is at least partially automatic, but can be modulated by attention under some conditions, such as when the faces are somewhat difficult to perceive. Other studies have used the N170 to ask whether faces are processed in a specialized face module or whether the same neural process is also used when people process other sorts of complex stimuli for which they have extensive expertise. So, consistent with a key role for expertise, these studies have shown that bird experts exhibit an enhanced N170 in response to birds, dog experts exhibit an enhanced N170 in response to dogs, and fingerprint experts exhibit an enhanced N170 in response to fingerprints, according to the study done by Tanaka and Karan in 2001 and Bussy and Van der Kolk in 2005. Developmental studies have used the N170 to track the development of face processing in humans, showing that face-specific processing is present early in infancy, but becomes faster and more sophisticated over the course of development. Studies of neurodevelopmental disorders have shown that the N170 is abnormal in children with autism spectrum disorder. So overall, the N170 example illustrates the precise temporal resolution of the ERP technique. ERPs reflect ongoing brain activity with little delay, and an ERP effect observed at 150 milliseconds reflects neural processing that occurred at 150 milliseconds. Therefore, ERPs are useful for answering questions about the timing of mental processes. Sometimes this timing information is used explicitly by asking whether two conditions or groups differ in the timing of a given neural response. The timing information can also be used in determining whether a given experimental manipulation influences sensory activity that occurs shortly after stimulus onset or higher level cognitive processes that occur hundreds of milliseconds later. For example, ERPs have been used to ask whether attentional manipulations influence post-perceptual memory and decision processes. Meanwhile, the ERP study with schizophrenia patients shows how different ways can be used to isolate specific ERP components that reflect specific processes. Second, it shows how ERPs can be used to precisely assess the timing of specific processes that occur between a stimulus and a response. So overall, the ERP technique is the best available technique for answering many important scientific questions, but it is not a great technique for answering others. ERPs are just mostly particularly good for assessing the time course of processing and determining which process is influenced by an experimental manipulation and identifying multiple neurocognitive processes. Another advantage of ERPs over behavioral measures is that ERPs can be used to provide an online measure of processing when a behavioral response is impossible or problematic. This is called the covert measurement of processing. In some cases, covert measurement is necessary because the subject is incapable of making a response. For example, ERPs can be recorded from infants who are too young to be instructed to make a response. ERPs are also used for covert monitoring in people with neurological disorders who are unable to make behavioral responses. So these are some of the advantages of event-related potentials. And there are many ERP studies and ERP components out there. So here's just a list of a few that are discussed in Stephen Luck's ERP book. I won't be going over all of these for the sake of time, but feel free to read any of these in more detail online. So just briefly, other interesting ERPs that I didn't mention relate to language, long-term memory, emotion, errors, responses, steady states, and so much more. So to wrap up this lesson, we learned that ERPs are the measured brain responses due to a stimulus, and they are useful in many interesting ways, such as answering many questions about certain mental processes. Next up, you will learn and explore some basic data analysis code of an ERP experiment using the N170 dataset in the second part of the Google Colab notebook. So just a side note, a lot of the code used here in this notebook is provided by NeuroTech-X. NeuroTech-X provides other sample codes for analyzing other ERP experiments and not just the N170 experiment. So feel free to look up other NeuroTech-X notebooks to find other free and useful resources, including tutorials to learn more about EEG data analysis. So I hope you enjoyed these lessons, and let me know if you have any questions.